now into the science. But before to say what we do and why we like it, I would like to pose the question and to make clear why it's important to have nanochemical analysis. Because, of course, measuring with a Nikon IR or a nano IR biomolecular processes at the nanoscale, chemical, mechanical resolution is cool, but it's also important to understand why it is important. And I will say the key point is that when we look at nature, the first things that we can try to do is to look at it, to investigate it. But often this is not sufficient because beyond seeing, we want to be able to play with this nature, to taste it, to understand what it is. And in particular, when we translate our curiosity for seeing into analytical methods, non-analytical ones, of course, we can first talk about single molecule microscopy. This allows to have nanoscale imaging, to look at the heterogeneity of biomolecules, but is chemically blind. This has been this challenge to understand beyond seeing what is in nature has been undertaken by a series of many analytical methods in chemistry that, for instance, like infrared spectroscopy, can give an idea what is the chemical identity and properties of our sample in an unconstructive way. However, most of these analytical approaches in chemistry, they have limited spatial resolution. So in our laboratories, our scope is to break the walls separating these two fields and to have nanochemical analysis beyond seeing. In order to achieve that, we use the multimodal capabilities of FM-based infrared nanospectroscopy. Being based on atomic force microscopy, this allows us first to acquire the morphology or the topography of a sample with angstrom resolution. And this is very easy if you will not be familiar with atomic force microscopy. Will be sufficient to think that it will work as a person would read in Braille, whereas not when he's blind. There is a sharp finger that is the tip that is feeling the interaction between this tip and the surface to retrieve this topography. Then, if we combine atomic force microscopy with infrared light, and we measure the response of the cantilever, where the tip is mounted at every position when we send IR light, exactly as before Qiqi has explained, we can acquire chemical maps and infrared spectra. Of course, if we consider also that this cantilever is a sensitive force sensor, together with chemical information, we can acquire mechanical information. So this means that infrared nanospectroscopy allows us to go beyond seeing and to understand nature at the nanoscale with capability to smell, taste its chemical properties, as well as to play with it nanomechanically to understand its properties. And as Qiqi mentioned, we were lucky enough to start very early with the technology. So now it's more than 10 years ago. And at the beginning, already in 2014, we proved that FM-IR was suitable to study biomolecules. And in particular, we were studying large droplets to prove that the technique was feasible to study protein structure at a very large scale. Then we have proven that at a much smaller scale, the technique is able to operate in liquid to measure again protein structure. We also understood, since measuring in liquid at the single molecule level is still quite challenging but feasible, that having methods to prepare the samples in a more accurate way for rare measurements would have been very beneficial. And so we combined the technology with microfluidics preparation methods. And finally, these advances altogether have brought us to the ability to measure the chemical signature of single molecules. Of course, these capabilities have been applied for a lot of scientific discoveries in the field of protein self-assembly, that we will look at it in particular today more, to study cells and tissues, as well as bio and non-materials, and to study biomolecular interactions. Today, I would like to give you a flavor of how we have reached single molecule sensitivity and how this has been key to study protein self-assembly and in orphan disease. So, as I just said, first part of my webinar will be about discussing how and we reach and why single molecule sensitivity beyond seeing. So to understand why this is so important, we should start first considering the following simple experiment. Let's suppose that we have in a test tube some protein aggregates, in a test tube some nucleic acids. As I said before, there are many nanoscale imaging methods that can have great resolution and image capabilities on these samples to resolve their morphology and heterogeneity. This is very easy when the two samples are separate in the test tube. But let's suppose to mix them in the same test tube and repeat the measurement again. What we will notice that in our map, doesn't matter how much is resolved, it will be now much more difficult to distinguish what is protein, what is DNA. Of course, as I said before, analytical methods can come in our help. And for instance, infrared spectroscopy can acquire spectra of our sample to show that both the peaks of protein and nucleic acids are present in our sample. But if we will use this technique to image our samples, basically we will not be able to do so because we don't have enough spatial resolution at the nanoscale. Infrared nanospectroscopy has solved this issue and the separation within these two fields, because together with a morphology map, we can now acquire a chemical map, where we can say, by looking at contrast, for instance, a fixed wave number, which sample is richer in a component or not in the infrared. However, we can also acquire spectra, so in a simpler way, we can point now our tip on the top of single molecules to see that, for instance, this is a protein sample, while the other one is a protein molecule, while the other one is a DNA molecule. So this means that we can, as such, do nanochemical analysis at a single molecule level. This seems simple, but in reality it's very difficult, and only in the last years we are being able to achieve this. Before even to attempt to measure our samples, we have to be very careful, because in order to measure at the single molecule level, we need to first preserve the conformation of our molecules on a surface that is needed to perform FM measurements that, moreover, are performed in air, because in liquid we have not yet reached single molecule sensitivity. For this reason, we have developed a